Don't type it all into YouTube. It's okay.
Okay, we're on YouTube now, so I guess we're ready to start. And it's a pleasure and a privilege to be introducing Amy. So Amy Brock is an associate professor. Sorry, I hear something. Okay, Amy Brock is an associate professor. I mean, I'm just reading out the slide here. The Department of Biomedical Engineering at the Institute for Cellular and Molecular Biology at the University of Texas, Austin. She's with the Center for Computational Oncology, which is a major department made of mathematical modelers, engineers, and biologists working to sort of use computational approaches in order to understand cancer. Amy has a bachelor's in biology from MIT, a PhD in biological and biomedical science from Harvard University, and she's still in Harvard to do a postdoc where she worked at the Children's Hospital, Boston Children's Hospital, using engineering approaches. She joined UT in 2013, so 9 years ago, and she's now an associate professor in the department where she's leading a group of researchers working on imaging and mathematical modeling, working with experimentalists in order to actually get beautiful models where she understands cancer as a complex system, trying to understand heterogeneity, and how to basically use cancer as a complex system in order to treat patients differently. So Amy, please, the stage is yours.
Okay, thanks so much. Thanks for the invitation to be here. I hope sometime we can do it in person, yes. But this is great for now. I really appreciate the opportunity to present some of our recent work to all of you today. And so one thing I was saying to David before, please, if you have a question partway through, just go ahead and unmute. I'd rather just take the questions as they come, so you don't sit there wondering, confused for a long time about something that I might have forgotten to say, especially because we have a group with a lot of different, backgrounds, and we all approach this from our different areas. And mine is, as David said, my training is really in biology, and I came from the experimental side to try to incorporate more quantitative techniques later on. But I know many come from the opposite direction as well. So I'm going to hide your faces. And I may not see if you virtually raise your hand, but please just call out whenever you'd like. Great. So let's just get this going here. Good. Okay. So our lab at UT Austin is really sort of broadly interested in understanding what are the functional roles of heterogeneity in mammalian cell populations, and we're especially interested in cancer cells, in their therapeutic response, and in the rise of resistance. And I think for this audience, I probably don't need to convince you too much that looking at variation, in addition to looking at different patient tumors or intertumor variation, looking more deeply at the variation among cells within a tumor is interesting and important, and we know has a lot of implications for disease progression and for treatment approaches. So, our kind of big goal in understanding the contributions of heterogeneous cell states is that we'd really like to be able to track individual cells as they go through the emergence of resistance. And this work came out, this was my postdoctoral work, so it's from a long time ago, and it came out the year that I started my faculty position here. In this study, we were also interested in that question, and we were able to track this process here using a fluorescent substrate for a resistance marker. So the protein we were interested in is called MDR1, multi-drug resistance 1, also goes by P-glycoprotein. And we had a fluorescent substrate to track when that protein is active in cells. We also had antibody labeling and so on. So we were able to do a lot. But an aspect of those studies that was really challenging and kind of bothered me was that you need to know some a priori knowledge of what is the resistance mechanism to do this, which is often not well defined in real world settings, right? And also likely to be changing over time. The resistance mechanism is probably changing with disease progression and with treatment. So the first couple of years of the lab, we really puzzled over this need for some kind of unbiased marker system that didn't need any previous knowledge of underlying resistance pathways to get started. And what we eventually came to was this approach that relies on labeling cells with random nucleic acid barcodes that really allows us to do the types of studies that we would like to do. And so this design was the work of Aziz Al-Kafaji, who's a PhD graduate from the lab, and we first published the method in 2018. And the way it works is as follows. So every cell in the founder population in a dish gets labeled, and that's using a viral vector to deliver the label. And every cell receives a unique random 20 nucleotide sequence that serves as the barcode. And that sequence in our design also encodes a guide RNA. So the nucleotide label gets integrated into the cell genome, it's heritable to all the daughter cells. And in addition, the cell is expressing a unique guide RNA sequence. And the 20 nucleotide random bit of the sequence is also a CRISPR recognition site in the guide RNA. So we'll come back to that a little bit later on. And maybe I should say the backbone of this vector, which is shown at the bottom here, is the CropSeq vector design that was first used by Christoph Bach at the University of Vienna. So with cells that get tagged in this way, we can do several unique types of studies. So first, what we are able to do in a cell population, we can measure the clonal composition of that population by performing targeted sequencing of the genomic barcode ensemble. So just taking the regions, flanking the barcodes, amplifying those and reading them by next-gen sequencing. Those counts, those read counts will correspond to the abundance of a clone in the population, right? Second, because the barcodes are express sequences, in addition to being integrated into the genome, we can read them out in single-cell RNA-seq workflows to give us information about lineage resolved or clonally resolved single-cell transcriptomes. And I kind of use these terms interchangeably, lineage and clone. And so you'll hear me say both of those in the presentation. My experience has been those of us who came up through the cancer field are used to talking about clones, and maybe if developmental biology is your background, you may have talked about lineages, but I'll use them both. And finally, we can go further with this technology, and we can use these guide RNA barcodes not just as a static label for quantitation, but also as a kind of actuator to drive gene expression in specific clones within the overall heterogeneous population. And that's a capability we'll come back to in the later part of the talk. Okay, so
a quick question. Yeah. So, traditionally those sort of barcoding approaches only tag a subset of the cells, right? Not all of them. Is this an approach that allows you to really have a barcode for every cell?
Yeah, so what we do, let's see, I think you can hopefully see my pointer. So at this step, these are meant to be viruses, right? So the cells are getting tagged with the virus. We have a fluorescent reporter that lets us know which cells have incorporated the label, and then we sort those. So we start with a fully labeled population by virtue of sorting out the cells that we verify are expressing the construct. Yeah.
So but this is not in animals, this would be in vitro.
This is in vitro.
Yeah.
Right.
So, okay, understood. Thanks.
Yeah, and so if your delivery system is less, like if there are some methods that don't use virus, they may use nucleofection or something instead. and have a low, a lower rate of labeling. We actually, maybe I'll say one more thing about this. We purposely keep the rate of positively transduced, that's the cells that pick up a virus, right? We keep the rate of positively transduced cells low by using a lower titer of virus, so that we don't get cells with multiple barcodes, right? So we assume Poisson statistics. We want many cells to have no label, and most cells to have only one. And then we sort out the ones that are positives, and that becomes our starting pool of cells. So a feature of barcoding that I think is really useful for quantitative studies is this ability to compare, behaviors of clones across, I don't know if I want to call them true replicates, but across replicates. So if we create replicates from the same starting population and we design them carefully so that they should each contain the same collection of barcodes, then we can ask questions like, across parallel treatments, or different perturbations, different drug treatments, and so on, do we see the same clones always win out? And if we see that, then that might indicate that there were some pre-existing resistant cells in the population, and that pre-existing resistance mechanism is really underlying survival in the population. And in contrast, if we see many different clones contributing to the outgrowth population, so many cells have the potential to survive treatment, that might be an indication that there's some de novo process happening, which could be mutation, could be some epigenetic change that broadly might be describing the resistance mechanism that's important in that scenario. So I really like this feature of barcodes. I think it makes me think of Gould asking, can you replay the tape? What happens if you replay the tape of life in macroscopic evolution? I think it lets us do something similar to track the trajectory of cells in a dish. So one of the first things that we did with this system was in collaboration with the lab of Kathy Wu at Dana-Farber. And we used this set of tools to examine clonal responses of CLL cells to cytotoxic chemotherapy. So CLL is a B-cell malignancy. It has rates of relapse rates of about 20% within 2 years. And we decided to focus on that relapse type setting to ask, what is the origin of the cells that survive chemotherapy in vitro, and also analogously in a clinical setting might contribute to the relapsed population. So Aziz from my lab worked together with Kathy Gutierrez, who is an MD PhD student in the Wu lab. And this was a really close collaboration. We had people going back and forth and spending time in each lab and so on. So it was a lot of fun. And they worked together to establish a barcoded CLL line from the HG3 cell line. And then they split that population, that labeled population, into 8 replicate cultures so that the same barcodes were represented in each one, and treated them with fludarabine, mifosfamide, cytotoxic chemotherapies. Mifosfamide is the active in vitro analog of cyclophosphamide. And they treated them at a high dose, about LD95, and then washed out the drug and let those cells regrow, which takes about 4 weeks. And then asked, what was the origin of these resistant cells by performing next-gen sequencing of the barcode ensembles. So the first thing that you see here is that the number of barcodes, there's a really severe bottlenecks. The number of barcodes decreases from time point 0 to time point 1.
Sorry, quick question. Is this truly resistance, or are you looking at the survivors? In other words, like if you are to repeat the treatment, would it still kind of eliminate a large fraction of the cells?
Yeah, so we did check that. We did check that later that the survivor cells are more resistant to the drug, and confirmed that that was true. So if that's, yeah, these are the survivors, but they do seem to have a higher, they have less sensitivity to fludarabine levostomide.
Okay.
So, and then looking at the heat map, every row there is the abundance of one barcode, one clone. So you can see many clones are sensitive and die off. Most survivor clones appear in multiple replicate populations. Another way to look at that data is as stacked bar plots. And what those show is that 30% of the clones were in common across all 8 replicates. And in addition, the last set of stacked bar plots shows that those shared clones also comprise the majority of the post-treatment cell population. So this was a pretty good indication that there was a strong contribution of some pre-existing resistant state already. There's both, of course, there's always both in the data, but here the pre-existing resistance mechanism seemed to really dominate across these 8 replicates. And I think I said 30% were shared in common across all 8, and the majority were shared across 6 or more, or across at least 6.
Amy, sorry, maybe you already said this, but, and this sort of relates to Andre's question, but how long do you treat for? How long is that bottle?
Yeah, so the treatment itself is 48 hours, and then the drug is washed out and the cells recover without drug in the media.
And is that for another 40 hours or?
No, until they regrow to the initial population size, which takes some weeks.
Have you looked at any of those intermediates between those?
Yeah, we do. I didn't put all of it into the talk, but in the paper, we also, I have a little bit of it here. We also took time points at the about halfway through the repopulation. So at day 12. Yeah, and we were able to find some interesting signatures there that inspired us to go back and look at the pretreatment. I'm going to talk about at least one piece of data from that time point. Okay, so we next wanted to characterize the gene expression profiles that are underlying these survivor cell populations. And this is possible with the 10x Genomics single cell RNAseq platform, because those barcode sequences are, they're integrated, but they're also expressed sequences. So we can use that aspect of the technology here. And so this analysis was the work of Eric Brenner, who's a senior grad student in the lab getting ready to defend in a couple months. And he performed these lineage resolved single cell RNA-seq analyses on 2 of the replicate populations, 2 of the post-treatment replicate populations, which are shown here in purple and blue. And then the pre-treatment sample, which is shown in green. And this data represents about 9,000 individual cells. It can they group into they group into 8 transcriptionally defined clusters. So the pre-treatment cells, those are the green, group into 2 clusters, and the post-treatment or survivor cells fall into about 5 overlapping clusters. And then because we're able to measure the transcriptome and also know its clonal family, its barcode identifier, on the rightmost UMAP here, you see labeled the 10 most abundant survivor clones. After that, we start to run out of colors, but there are 1,050 different clones in this data so that we can read out the 20 nucleotide barcode identifier. So just about all of those gray dots, we also know their barcode ID that's associated with that cell. And so with this kind of data now, we can do something new. We can trace the trajectories of clones and follow them, right? Using the barcode identifiers, follow them from pre-treatment to post-treatment. And when we do that, what we learned is that all of these abundant survivor clones in the, I'm going to point with the mouse and hopefully that will work, in the large grouping, which comprises 95% of the survivor cell population that regrows, they all originated from this single small pretreatment population. They all came from cluster 5. And all of the clones in cluster 6, which is a minority of the survivor cells, a little less than 5% of the survivor cells, they can be traced back to have their origins in cluster 2, which is the majority, is about 80% of the pretreatment population. And there's almost no overlap. Clones don't crisscross and the same clonal identifiers aren't found in more than one of these. So we're really seeing, and this was pretty surprising, I thought. It's counter to what I would have guessed. We're seeing these 2 discrete gene expression states in the pretreatment population, one composed of a subset of clones that we call have high survivorship potential, and another composed of a different subset of clones with low survivorship potential. And their transcriptomic signatures are really sort of preset before drug exposure and linked to their clonal identity. And so, like I said, almost no cells deviate from these 2 possible trajectories.
So it's kind of very interesting. So these cells are kind of that give rise to the surviving and resistant cells. They're not forming any kind of clearly distinct sort of cluster, even though you see very high reproducibility in these biological replicates that the same kind of clones are surviving. Is this correct? And another sort of trying to understand the data, it looks like there's surviving cells, resistant cells change through the treatment phenotypically, so that it's not that there's a simple selection of something that's pre-existent. Are these 2 statements correct? Or
so the Leiden clusters, I mean, you can always find clusters, right? So there are transcriptional differences within within each of the pre and post treatment populations, where you can break down this large cluster into potentially 5 different subsets of transcriptional profiles. So there are transcriptional differences, but in terms of how they respond, in terms of survival, in terms of phenotype, it seems like they have these 2 trajectories only,
yeah,
right? So those 5 transcriptional states, are kind of, at least in terms of survival post-treatment, are equivalent in that scenario.
But there seems to be both kind of induction and selection, not just simply selection, that you have post-treatment, you have the same phenotype as before treatment, right?
Right. I mean, there definitely is a change, right? There's no cells found in cluster 2 in the post-treatment, and no cells that still occupy cluster 5 in the post-treatment. So there is definitely a change.
Yeah, very interesting.
For sure. Yeah. So we did marker gene analysis, all the sort of typical single cell RNA-seq things to do, and saw that this high survivorship subpopulation at time point 0, so pre-treatment, had upregulated Wnt signaling, CXCR4, other marker, JAK1, other markers of chromatin modification. I know the heat map is kind of small here. The low survivorship subpopulations were marked by upregulated some adhesion receptors. ITGB2 is one, which is also CD18, FGFR1, and some other antigen presentation molecules. I think from the heat map, even though it's small, even if you squint, it's kind of clear that both the high survivorship and the low survivorship profiles are kind of well maintained pre and post treatment. So there's a lot of stable features. In addition to, I think as Andre just pointed out, there's changes as well. What was really neat was with our collaborators at Dana-Farber, we were able to look at patient samples and do analysis of matched transcriptomes that they had collected from patients before and after relapse to fludarabine-based chemotherapy. So we had a cohort of 23 CLL patients, And there we were able to confirm an enrichment of the high survivorship signature in those patients that experienced relapse, but not the low survivorship signature. And specifically within that signature of CXCR4 and Notch signaling that were enriched at relapse in that patient cohort. Wnt did not reach significance there. So to learn more about these different responses of the high and low survivor cell, subpopulations, I'll just sort of quickly summarize a little bit of this work. We looked at earlier time points in the recovery from drug treatment. So this is the intermediate time point that we collected at day 12. So it's labeled as time point 0.5, but it's halfway between the pre-treatment and the post-treatment time point. And we looked there out of sort of curiosity of how the cells are responding during this recovery period. And we saw that both the high and low survivorship cells at that time point had upregulated apoptotic stress responses. I think made a lot of sense. There's still continued die-off over many days, even after the drug is washed out, right? And as those populations are starting to rebound. And then we looked back, that inspired us to look back to the pre-treatment populations and see what that signature looked like earlier. And we saw that the low survivorship subpopulation was really more primed for apoptosis, even at pre-treatment. And that's reflected in this heat map in the middle. The differences in high survivorship clones at pre-treatment and at the intermediate time point by GSEA analysis also revealed that there was a oxidative stress response, DNA repair pathways were upregulated. So maybe an indication that a lot of genes involved in some of these DNA damage response pathways were already expressed at higher levels in the high survivorship versus the low survivorship subpopulations, and maybe potential priming of that DNA repair pathway in those cells. And then a study that we added later on was to look at apoptotic priming by BH3 profiling. And this is actually was separated out cell subpopulations. And here, we also saw that the low survivorship cells were much more significantly primed for apoptosis in the pretreatment population compared with the high survivorship cells. So a lot of this data ended up in the supplement, but I think it's some of the most interesting data in the paper. I think that's kind of a really compelling bit of the story. So I wanted to include it here for you.
Amy, sorry, on that point, do you think that is really kind of, in some sense, illustrating the escape route from the drug?
Yeah, I think so. I mean, for sure, there's, it sort of hangs together. And I think we don't know necessarily, upstream of that. We haven't linked it to a particular genetic or transcriptomic or epigenomic change, but it certainly looks like at the functional level, this fits well with what we've seen. And
that, remind me again, what was the drug?
This is fludarabine and lefosfamide. So it's a cyclophosphamide. And
so
they're cytotoxic.
So they're not targeted. They're more,
Right,
okay.
That's right, yeah.
Because that DNA repair is kind of interesting.
Yeah, I think so. Mm-hmm.
Yeah, thanks.
Did you see anything where did you do anything where to look at like how they might move between these two groups over time? For example, if you took the high survivor population out alone and reculture it, do you get low survivors in the majority after a few weeks or whatever?
So we don't, and I'll show you some data where we separated them. We didn't see signs of that kind of repopulation of the parental distribution in this model, but we do have some separate long-term co-cultures, and then what I'm going to come to in just
Cool,
a couple slides is co-cultures. So actually, it's just coming here. So that's perfect. Thanks, Mark. Right. So it was, just even without the drug response, it was really sort of intriguing to us that there were these two subpopulations. They coexist. They've obviously been in this, in this cell culture model over many decades since it was first started. And their proportions are stable over time. So this what we're calling the high survivorship subpopulation is the minority of the culture, and the rest is this low survivorship subpopulation. And so we were really curious to learn if they interact and how their growth rates compare, and just some of these basic properties of these two subpopulations. So one of the things we did first, because we have the barcodes there, was to measure whether there were differences in growth rate of these two subsets of cells in the parental, in the bulk population. And so to do that, we took barcode sampling data by next-gen sequencing, just during normal growth at 3 time points, 48 hours apart. And then with that, we get information about the proportion of the total population that each lineage occupies, right? And so combining that with measurements of the total cell population, we could estimate the number of cells in each lineage at 3 time points. So I'm trying to be really explicit about that, just because some of you who might think about how to use this type of measurement as modelers, right? So there's a little bit of there's some steps in between counting cell number of each, carrying each barcode, and the type of next-gen sequencing read data that one gets, which is % of the total population. And so we used the barcode sampling there, and then we took, we did sorting based on CXCR4 and CD18, which were 2 labels that well separated these populations, and assigned a lineage to either be part of the CXCR4 positive population or the CD18 positive population. So that's how we assigned lineages to either high or low survivorship categories. And when we did all of that, we found that we really didn't see any differences in growth rate when these lineages were all together in the bulk population. And so, yes, sorry, I should have showed you this. So here are the 2 labels that we used to separate the high and the low survivorship populations out by flow cytometry. So when they're together, we didn't see any difference in growth rate, but if we sorted on these and then cultured the populations separately, it was pretty clear that the higher survivorship subpopulation had a faster growth rate than either the bulk or the low survivorship subpopulation. And then what we did was to label the subsets of cells with fluorophores, so we could co-culture them over time and track them in different proportions. And we did that with a range of different starting seeding proportions. And we monitored those cultures over 2 weeks, 14 days. And what we found here was that if the population started with only 10 to 20% of high survivorship clones, which is what the cell line, the parental cell line also displays, then it had a stable population composition over time. But if the initial abundance of high survivorship clones, if we seeded that beyond 30%, then their frequency started to rise over time, although still not at the rate at which the high survivorship clones grow when they're on their own, right? And so these findings, really point to the idea that the low,
So can I ask, is this in serum conditions?
All the normal growth conditions,
Yeah. Because I just
normal growth media.
One of the things about cost is that maybe you don't pay it if you've got an abundance of I don't know, resources. So I wonder if, if you were to use, say, like, I don't know, some sort of conditioned media that was maybe a little bit more deprived, would there be a difference?
Yeah, that's a great idea. I don't think that we did that with these cells. That's a great idea. Yeah, I'm writing down. So what we think here is that the low survivorship clones impose some anti-proliferative effect on the high survivorship clones. We were not able to confirm a biological mechanism that would underlie that interaction. We did identify some candidate ligand receptor pairs that are predicted from the single cell data. And some of those, especially at least one of those, the LGAL and SORL1 pairing have also been noted as in patient data. So that's kind of interesting. We tried a few different blocking studies, and during the review of the paper, we just didn't have time to really hammer that out in a way that was convincing to us. But we surely think that there is some kind of interaction present there that maybe will be a future follow-up. And one thing that I realize now, I should have told you earlier about these cells, is that they're suspension cultures. So this is even more intriguing, because we've seen things like this in 2D adherent culture. And we really thought that in suspended cells, there would be, the cells would behave in a little bit more cell autonomous way. But the other thing I can say about that is that even in suspension, sometimes cells are clumping together. And so this is a place where we might look more deeply in the future. So I said that our barcoding system has another functionality that we haven't really revisited yet, that lets us evaluate subpopulation heterogeneity with greater resolution. So let me tell you how this part works. So now that we know that there are some clones that are particular interest based on their fate or their high abundance or so on, we can physically isolate them out of the bulk population from some archived samples at different time points, pre or post drug exposure, using this ability of guide RNA sequences to drive transcriptional activation. And so this aspect is sort of very unique to the design of our system. And the way that it works is by delivering to the whole population, the whole labeled barcoded population, either by nucleofection or transfection of the population, this gene circuit. It's a very simple gene circuit. It consists of the Cas9 transcriptional activator variant, which is here the one we use is dCas9 VPR, plus a reporter plasmid where we place the barcode that corresponds to our target clone, the one we want to isolate, upstream of a minimal promoter and a fluorescent reporter. So this basically sets up this simple kind of logic gate in the cell population, so that only the cells that express the matching guide RNA barcode are able to recruit the Cas9 transcriptional activator to the promoter site and drive GFP expression, and then those can be collected by fax. So sometimes when I am on grant panels and so on, I hear the caveat that single cell omics is somewhat limited because of the destructive measurement, and you only measure one cell and then it's gone. And that's certainly true. We can't make parallel measurements on the exact same cell, but what we can do is you'll go back to a sister cell or grandparent cell or however, another cell in the same clonal family and do some complementary types of analysis. So that's sort of the power of this technique. So in the CLL cells, to look at subpopulation heterogeneity with a little bit greater resolution, we invoke this ability to do clonal isolation, and we isolated out 2 clones that were present at high levels in the high survivorship subpopulation. And just to make our lives a little more complicated, I think when we isolated them off the sorter, we also seeded them into individual wells so we could get subclones. So we have major clones now and subclones, which may be, a little too much, but that's how it was done. So we isolated these two lineages, given that the clonal families, that we've been looking at had these consistent phenotypic responses. One of the first things we did was to look at to look and see if there were differences in SNVs by whole genome sequencing of those lineages. And we used here the low survivorship subpopulation as a reference standard. What we found was that between the two lineages, out of that population, about 40% of mutations were shared, which indicates they likely have a common ancestor, but most were unique to one clone or another. And then the last panel shows you that after exposure to the chemotherapy, there's increased diversification of each of those lineages, which I think makes good intuitive sense and is also reflected in clinical samples with elevated mutational burden after in relapse from chemotherapy. And then on the left, this table shows 33 mutations that were predicted to have some functional impact, given that they're missense mutations with elevated polyphene scores. So they're either nonsense or splice site mutations and so on. And one of those was a p53 regulator SHRM-L2, which has been associated with apoptotic response to oxidative stress and is upregulated in other tumor types. So that's one that we might like to follow up on some more. For each of those two major clones, I said we looked at 3 subclones. Sort of this was just driven by curiosity at this point. Each of those subclones, among the subclones, there was about more than 70% of SNVs were shared, but about a little less than 30 were unique, and that means they're acquired during the expansion of the population after labeling from that initial barcoded founder cell. So that was a little bit, I think, of a surprise to a lot of us. And again, after treatment, there's increase in the acquisition of new SNVs here too. So overall, what did all of this sequencing told us? I think that the high survivor subpopulation, it's made up of numerous genetically distinct lineages. We only looked at 2 for reasons of limited resources. The unique SNV signatures of the subclones, they're getting maintained over expansion, but they're also constantly diversifying, both in the absence and the presence of this strong environmental pressure that we applied. So I think that was some interesting baseline information for us about how subclones within a cell culture are responding over time. Given that
quickly
there's,
ask Amy.
Yeah, go ahead.
So if I'm understanding that right, are you saying genotypically they're diverse, but phenotypically they're similar?
Right. Yeah. And these are just 2, right? Just 2 clones. They're also the ones that survive this pretty extreme bottleneck and are abundant in the post-treatment survivor cell subpopulation. So maybe that's not, so surprising, because we picked them based on similar phenotype. And they're, maybe at the outside range of what the cells are able to survive, but that's exactly right.
Yeah, but it's nice to see some sort of, it's like a convergent evolution that's occurring. You are giving them an extreme pressure, but there could have been a mutation there that gave them everything they needed. But I think the point here is evolution acts on the phenotype. And so there's multiple routes to that phenotype. And probably if you had enough money, you could probably find a few more.
Yeah, absolutely. I mean, I think that would be so neat. And also the space that they're exploring, even without, pre-treatment, without any pressure, seems to be pretty significant, right? There's a lot of changes happening. And I'm going to talk about transcriptional states in a minute, but this is, I think it's really neat to know sort of baseline properties of what these cells are doing. Yeah, so given that there's this high degree of genetic variation among clones that behave similarly, we also wanted to look at transcriptomic heterogeneity, and we did single-cell RNA-seq on these, so it's 6 isolated subclones from pre and post-treatment. So we hashed and we ran 12 samples together on one channel. So this gives you about 500 cells per subclone to analyze. And we found that this tree, this dendrogram, really just shows that the cells largely segregate based on their treatment status and then their lineage identity. So kind of recapitulates their clonal relationships. And then we asked whether, could you classify an unknown cell by its lineage identity by using this data. And so here Eric Brenner, again in my lab, used supervised learning approaches, and he generated a multivariate classifier to learn the transcriptomic signatures of these 6 different subclones within each time point. So here you see the pre and the post-treatment separately. And those models were highly accurate with weighted precision scores higher than 0.97 for both of them. So there was really robust transcriptomic variation within those 6 subclonal populations. And then that sort of emboldened him, I guess, to look back and ask whether these clonal transcriptomic signatures are, we're seeing that they're characteristic, are they stable over treatment? So he generated a new model looking back at the 10 most abundant survivor lineages from the first set of single-cell RNA-seq data that I showed you, and tried to classify those by their lineage identity. So this is now a little bit harder 10-class model. And he ended up there with weighted precision scores of 0.73 for the pre-treatment, which is the more diverse population, and 0.882 for the post-treatment population. But again, supported the idea that lineages have these stable transcriptomic signatures that are even conserved over the treatment course, right? I think this last heat map just shows that ranking genes by their weighted coefficients and visualizing expression of the top 20, there's not any single, there's no single gene that's this expression value explains, each lineage characterizes a particular lineage. So it's really a composite across the transcriptome. I think altogether, these kind of observations are, linking the accumulation of genetic alterations to transcriptomic variation, and then the development of characteristic subclonal signatures that they get maintained during normal cell expansion and during treatment. So it's, this peek at the balance that's happening in cell cultures. I'm not sure how we do on how do we do on time, David? Should I go a little bit longer, or so I have another section?
Longer, yeah.
I can, yeah. Okay. So let me summarize this section and just say that express barcodes let us track trajectories of cells that are responding to therapies in HG3 cells, CLL model, one that has a lot of similarities with the mutational spectrum in patients. We were able to look at and identify these 2 discrete subpopulations with different drug sensitivities, different transcriptional programs. And this approach is sort of a way to start to quantify the balance between genomic diversification and transcriptomic stability, and dissect some of these complex clonal dynamics, which are, even in cell culture, I think more complex than we probably appreciated when we started out. I was going to show you some unpublished data for a few minutes, which has sort of a very similar flavor to it. You can see I reused the diagram to tell you about a similar study with triple negative breast cancer cell model, just as a way to talk about some of the similarities and then some of the differences that we're starting to see. So in this study, We treated MDA MB231 cells, just sort of a workhorse triple negative cell line, with doxorubicin, again at a high dose for 48 hours, then washed out and let the cell population regrow. I don't have all of the replicates here because some of this is still in the works. But if you look at these stacked bar plots that we got from performing next-gen sequencing of the barcode populations, what you can see is, again, there's some similarities, but it's kind of more of a 50-50 situation. Like, many barcodes are shared across all, but many are also unique. So we'll see how that plays out with more replicates, but it's a little bit different scenario with compared to the CLL population, where there was this really strong signature of a pre-existing population. And that's sort of also comes out when we do single cell RNA sequencing analysis. So I'm going to show you some UMAPs of 4 samples. So again, there's going to be a pretreatment population. The color scheme is different. Sorry about that. The pretreatment population is going to be in purple. And then 2 treated cell populations of the survivor cells at the end of treatment, and those will be blue and green. And we also here collected a time point kind of acutely 30 hours into the treatment to see how the cells were responding. This is before there's a lot of cell die-off. There's not really a big change in viability yet, but the cells are, certainly responding to this high dose of drug. And again, we can layer on top of the transcriptomic profiles the lineage information. And again, we have barcode identifiers for about 91% of the cells. And the rightmost panel here is just showing you the most abundant ones. These clusters that look like they're all red, there's actually other colors in there. It's just there's some very dominant survivor, like super survivor clones in these populations. So what have we learned from this so far? I think one thing that some people are surprised by, but others have also reported this, that the 231 cell model, many of us use it as an in vitro model of TNBC. It breaks down into these 3 different gene expression states. And it turns out that which are circled here with the dashed lines, and it turns out the clonal composition and then the fates of cells in each of these clusters is pretty different when faced with doxorubicin. And I'll just sort of go through a little bit one by one. So the cells that start out in this particular transcriptional cluster, they have a characteristic response to therapy that we're still a little bit working out what happens at the orange acute time point. And then they, after 30 hours, you can see they're never seen in the post-treatment sample. So they all go extinct. All the cells that start out in this pre-treatment cluster die off. And the cells in this cluster, which is kind of really dominated by just a couple of clones, especially this one abundant pink clone, they have their own unique trajectory of response. So at 30 hours, they move to a sort of specific transcriptional state, and then they comprise a minority of the long-term survivor cells, but they have a cluster that's represented post-treatment. And when we look at, sort of underlying gene expression signatures there, they have a lot of signatures of a P53 response and a couple of other markers that have been identified with beta-catenin and ESAM and SPDEF, which are other markers that have been associated with signaling in breast cancer and in other studies. So it's sort of an interesting one, and we're really kind of curious to see if this clone comes out again and repeats. We're doing biological repeats now. And then all the other survivor clones, except for that pink one, all the other survivors that comprise the blue and the green sample of post-treatment cells, they all start out in this cluster prior to any drug treatment, and they go through this set of potential trajectories in response to exposure. But being in that state pre-treatment isn't sufficient to confer survival. So many cells in that state, many clones in that state also die off. So we're starting to investigate that more. But I think that kind of fits with our targeted sequencing data, where we can look at many, much larger population, in that the signature of a pre-existing state is, it's not as strong, it's not as consistent in this particular model with this particular perturbation.
So Amy, is that a temporal trajectory you're showing there? Is that being inferred, or is that actually from different time points?
So this is from different time points. So pre-treatment here, post-treatment in orange, 30 hours in orange, and then the regrown cells in each of these two last populations, which is about 4 weeks later. So
3 time points.
It's 3 time points. Yeah.
So that's how you can see that trajectory.
And then, this goes back to Sandy's, point from earlier. What are the different ways that cells find to survive? So we can look at these two. Clearly, they're the post-treatment. The cells that make it through occupy different states, different transcriptional states, and so we can compare them. The one that's bolded here, those cells are marked by upregulated RAS and Rho GTPase signaling. The ones that occupy this other survivor state are marked by changes in ubiquitin-mediated proteolysis, tRNA biosynthesis, so some more kind of metabolic type changes. And one of the things we wondered is, could we look at the pre-treatment cluster and see any indications of those particular clones, just pinpointing those clones that end up as survivors. So we know the future survivors and the future non-survivors. And could we look back at that pre-treatment transcriptional state and, and ask whether there were any early indications of which strategy was a cell going to undertake to survive. When we looked there to see if any of these signatures are already changed in those rare clones before treatment, we really didn't see anything convincing about differences between the future survivors and non-survivors that correlated with these particular resistant signatures. But we did see some changes, at 30, it's here at 30 hours. And so when we look to the sort of acute response, we might be able to see some differences there. So that's another aspect that we're really exploring. It does look like by the time they get to the 30-hour cluster, some of the red ones are already showing signatures of upregulated ERK and Rho GTPase and MAP kinase signaling, and some of the and the yellow cells and the pink cells and so on that are going to eventually end up in this state some weeks later, maybe already at 30 hours, have some indications of changes that look like they're going in that direction. So that's something I think really intriguing that we'll be following up on before we solidify this study. For sure, these different pretreatment clusters that exist in the 231 cell line, they seem to have interactions. And I'm not going to talk about that today, but some of you have seen D.D. Gardner's work, and that's expanding. And hopefully, in a few more months, we'll have a longer story about how these different 3 subpopulations, even in the baseline cell culture, without any drug exposure or other perturbations, might be interacting with each other and influencing each other's growth dynamics just at baseline. So that's kind of a coming soon aspect of this study. So yeah, I think I could just close by a few thoughts about how to use this type of data and what it opens up. So I hope that you are convinced that adding barcodes kind of really expands what you can do with a lot of current workflows. They're compatible with cell lines. They're also, we're using them in patient-derived cells in vitro. And they really enable the study of clonal phenotypes, but preserved within the context of the heterogeneous population, which our studies are at least are starting to indicate that, taking clones out and studying them separately, they have very different behaviors than when they're in the heterogeneous population. So, and that's probably important to know. I think there's a lot of potential for using these barcoded collections of cells to facilitate sharing across labs. One of the things we've been able to do with the 231 clones is send the same library to a group that does measurements of nuclear force and apply physical forces to cells and look at response. So we'll be able to know, does clone A, those cells are like much stiffer in your particular specialized assay, and they're resistant to doxorubicin, and they have this transcriptional signature that we measure in our lab, and so on like that. So I think that's a really neat aspect of having labeled collections of cells. I think what we're starting to see is that these barcodes kind of expose some overlooked structure that underlies in vitro models that maybe arose during the history. It's really made me think a lot about how different, some of you have isolated cell cultures, and there's all different ways that they came about, right? So sometimes you dump, you take a tumor and grind it up. I've done this, and take a mouse tumor, grind it up in a dish. You get millions and millions of cells, you grow them, that's great, they adhere. Sometimes there's a small population of cells out of a larger diverse collection of cell types, and you just isolate those few hundred cells using an O-ring. That was an old style way to do cell culture. It kind of like put Vaseline down and an O-ring around the cells you want, and you just tryptophan those. So the history of how we got every cell line is a little bit different, and probably important, and there's probably signatures of that we're coming across now when we look at subsets of cells and how they behave in that model, which I think is really interesting. Some people maybe worry them about using cell lines, but I think it's fascinating. And I think for every cell library that we make, we're going to have to learn about what I'm calling its transcriptome diffusivity, which just means how stable is that cell's transcriptome over generations, and how long can we use a barcode as a good proxy for transcriptome and for phenotype and so on like this. So I think we're going to learn a lot about stability of cell states and stability of underlying transcriptome networks by using these tags. So a ton of different things to do. I think I will now stop here. I really want to emphasize that, especially the CLL study was a very collaborative project with the Wu Lab, and it was really driven by the graduate students. So it was all graduate students as lead authors on that project. And this is a view of the current group and some of the recent citations that are relevant for the barcode technology. And we have a lot to do, and we need more people to help us do it. So if you're interested or someone, please spread the word about positions that'll be available in our group. Thanks.
Great. Thank you very much, Amy. So I know people have been asking questions through, and obviously it's been a very animated conversation, but I also know some people were asking questions on the chat window, and I think this is a good time for those people to actually voice their questions. So I'll just go through the list I got here, and if the people who asked the questions want to come forward, I think that'll be much better. Freddie Winding, are you around? His question, but obviously a lot of these questions are context relevant. So if we don't have the slides, it's going to be really hard to figure out what those questions meant. Anna, are you around? Anna Miller? I will see.
Okay.
Question from Audrey.
There's one question about the sampling. Okay. I'm scanning through. I don't know exactly what point that was, but I could speak to. There's a lot to think about sampling because of the single cell RNA sequencing. So what we have learned a lot about experimental design, I think, as a result. So, in the next gen sequencing studies where we quantify barcodes, you can be very quantitative, and you can be really, you can do a lot of reads, right, to sort of exhaustively search for which barcodes are in a population. I mean, if you want to spend, a bit on sequencing, but it's not technically too challenging to get to the point where you're sure that you have measured just about every barcode in the population. And we did that by taking different numbers of reads and looking for when it plateaus off, when we don't identify more barcodes with more reading of the next gen sequencing. But then you come to, so that data is a way to be really quantitative. But then you come to the single cell RNA sequencing and all the UMAPs I showed you and so on. And of course, you're only running through some thousands of cells. I think when we started the barcoding, we were really excited to we'll put in, 1 million barcodes or whatever, because the power of generating random sequences, is really exciting. But what I advise to people now is you usually don't need 10,000 unique labels to follow all the clonal behaviors that you're going to see, or 1 million you certainly don't need. It's overkill. So we're starting to work in a range where we have about 1,000 labels, and you can see, that often they break down into 2 or 3 phenotypes at the 30-hour time point treated with doxorubicin. There's something like 6, but it's not thousands. So I think there are the sampling effects are important to take into account when you do these single cell studies, but we can still be more than enough labels to see the phenotypes of interest and the transcriptome states of interest, I think.
Very cool. So does anyone else has any question at this stage?
I do. So thanks, Amy. It was great to finally see it and hear it fully be presented, and I really enjoyed it. I think it's a really cool tool, and I'd love to see us using some of that. I know that we've been looking at barcodes and we've been thinking about transcriptional states. And my question really relates to that in terms of, the point you made about the pre-existing states that might be there may kind of be a throwback to the fact of how these cells came out of patients and were kind of selected and cultured over the years anyway. But thinking about how we then relate that to patient samples, and I know you mentioned that you did have some patient samples that I guess you took some feature set of your transcriptional profile and sort of looked at that on a sample, right? But you obviously can't have the entire profile. You have to take a subset of that. But was that insightful? Did it give you sort of useful insights?
Yeah, so I think I'm not able to answer that yet. What we have for patient samples right now, we're working with Dell Medical School, which is here in Austin, and to look at a colorectal patient samples, and many of them have been through PDX, which people hope preserves more of the heterogeneity. But now we do have some that are isolated straight from patient and not gone through animal models at all. So I think this is a way we can start. And of course, there's going to be patient to patient variation too. We can start to compare some of those differences. I know we're being recorded, so I don't want to say anything about PDX models that's going to get me in trouble with most of the community. But I think a lot of people hope that they preserve much more of the variation, but there's still, it's certainly, I think, fair to think there's a selection happening when
it's a unique environment,
animal, right? Yeah.
No, I agree. I think that's what kind of intrigues me is obviously in some sense the patients, that's the ideal ground truth, but we can't access that until after the fact sort of thing, or maybe during treatments. But then we need these tools, but they're models of that reality. And so, it's almost like the techniques that you're building are going to allow us to back calculate in some sense, or go back to the patient eventually and understand what we gained, what we lost, what's important there, and what, you know, should we really worry about this in reality because it never happens in a patient, for example. That's the sort of information that would be critical, especially as we start thinking about, can we exploit this therapeutically, which is my immediate obvious thought when I see this, is like, well, now we know what the resistance cells are, the mechanism they're using, and that, you know, that they're quite stable. Can we sort of go after them with a different strategy?
Right. Yeah. So I have lots of thoughts about that. And, you know, so one thing I think that it's actually, you know, it may be a real benefit that cell lines have this underlying heterogeneity. We, you know, I think some of us are not surprised and some people are very surprised by that. But it may be in a strong advantage. Certainly, these collections of cells, you know, found a solution to survival in a dish. And so that may be, you know, reflective of some of the survival strategies that they use in tumors as well. So I think there's potential there. One thing I could say about, you know, going back and forth with these labeled cells and then looking to see you know, how the same subsets respond in patients, which is something I really want to do. When we put the cell, we and many others have found when you take the barcoded collections of cells and put them into mice in an effort to do like a xenograft model, often what obscures that intermediate step is the take rates of different clones are very different, and they don't necessarily recapitulate what happens in a dish. And we don't know if that's, you know, if that is a closer or a poor proxy for what happens in patients. And I've talked to some other, you know, people who have used different barcoding systems who have found the same thing. The starting cell population that you may have your labeled cell collection, put that into a mouse, and it's not very easy to predict which ones are going to give rise to tumor in a mouse. And let alone, you know, response to treatment and so on. So I think going through the animal as an intermediate step right now seems like a really hard strategy. But what I'm more interested in is trying, you know, to see how far we can get with pushing these models in vitro, and are they at all an analog for what happens clinically?
Makes sense. Thanks.
Cool.
So one quick question. Fantastic talk, but the question is more practical. How do we get access to these tools? Are these publicly available, or?
Yeah, one thing that we kind of got slowed down on in the last year was getting them all onto Adgene, which was totally just institutional forms and things like that. But we are trying to get everything there. But in the meantime, I've shared them with a lot of labs. Just you know, if you send me a message, we have protocols and we have the vector library we've given out to a number of people, and even, you know, I'm also happy to share the labeled, like already labeled cell lines. So we have probably a dozen or so different cell lines, you know, different collections even of, you know, so many labeled 231, many labeled of other cell lines, and I'm happy to share those as well.
Great, thank you.
Cool. Once again, thanks for everything, Amy. It was a great talk and clearly sparked a great conversation.
Great, thanks for the invitation. Yeah, I look forward to continuing.
Oh yeah, and
lots of you about this down the road. Yeah, appreciate that.
Cool.
Thank you. Mark, did you have a question?
No, I just said very nice talk, very interesting.
Okay. Cool.
Thanks, Amy. Really nice talk.